Good evening. Welcome, everyone, and thank you for joining us tonight. I'm David Dodick. I'm Chair of the American Brain Foundation Board of Directors. And tonight we're excited to have you here and excited to have a very special guest speaker where we'll talk about an exciting area in neurology, gene-targeted therapy, specifically for amyotrophic lateral sclerosis. Our foundation is committed to sharing important resources and increasing public awareness around brain disease, and our webinar events, which have become quite popular, like this one tonight, are opportunities to connect you with experts in various topics of importance and interest around brain diseases. The American Brain Foundation, as you know, funds research across the entire spectrum of brain diseases because we know that brain diseases are interconnected biologically many times, and curing one disease may mean that we will have a cure for many. So let's move on to our topic of conversation for tonight, which is gene-targeted therapy. And as researchers pursue treatments and cures for brain diseases, gene-targeted therapies have emerged as an area with the potential to provide solutions to some of the most devastating diseases we face. And of course, one of those is amyotrophic lateral sclerosis, or ALS. We are actually at the American Brain Foundation now currently funding multiple genetic-based projects, including 2 researchers studying gene-targeted therapies, specifically for ALS, and 2 researchers looking at genetic risk factors in ALS. So today's discussion will be centered around how genetics research is resulting in potentially innovative and important therapies for brain diseases in particular, in general, but in particular for ALS. So I'm delighted to welcome our guest speaker tonight, Dr. Timothy Miller. For more than 2 decades, Dr. Miller, one of the nation's foremost researchers in neurodegenerative diseases, has been searching for better ways to treat, prevent, and cure ALS. Currently, Dr. Miller holds an endowed chair, the David Clayson Professor of Neurology at Washington University School of Medicine in St. Louis, where he leads the Miller Laboratory and the ALS Center. He directs research into innovative treatments with a focus on therapeutic strategies that turn off harmful genes in the brain and spinal cord in people with neurodegenerative diseases. Dr. Miller provides clinical care as a practicing neurologist as well. He serves as the principal investigator on numerous clinical and research studies and directs all of the research taking place in the Miller Lab. In 2018, Dr. Miller received the Sheila Essey Award, which is an award given by the American Brain Foundation in partnership with the ALS Association and the American Academy of Neurology, which recognizes significant and substantial research contributions in the search for effective ALS therapies. Dr. Miller's work continues to improve the lives of those diagnosed with neurodegenerative diseases like ALS. We're very excited to have him here. Dr. Miller, thank you for taking the time and speaking with us tonight. Welcome.
All right. I am absolutely delighted to be here. Can everybody see my screen and hear me okay?
Yes.
Okay, very good. All right, so I'm going to jump in. I always start with my industry collaborations, in particular Ionis Pharmaceuticals, with whom I've had the pleasure of developing antisense oligonucleotides, and I'll talk about some of those this evening. And also Biogen, with whom I've had the pleasure of running clinical studies, and I'll talk about a Biogen-led clinical trial. We were talking about ALS. I think many of you are familiar with ALS or Lou Gehrig's disease, but I'll just hit some of the highlights, make sure we're all on the same page. It's caused by the loss of motor neurons, those cells that connect from the spinal cord or from the brain out to the muscles, or dysfunction in those motor pathways in the brain. And the loss of those cells, the change in those cells, leads to weakness and stiffness, and that occurs in the arms and the legs. It also affects swallowing, speaking, and breathing. Unfortunately, death typically occurs 3 to 5 years after the onset of ALS, and typically from respiratory failure. There are 3 FDA-approved drugs that I won't talk about tonight. Each of them has a clear impact, but it's minimal on the slowing of disease. And I'm going to talk to you about a novel therapeutic strategy for a genetic form of ALS. Most ALS is not genetic, or at least doesn't run in the family. It's a singleton or sporadic disease, but approximately 10% is familial or runs in the family, often a dominantly inherited disease. And if we drill down on that 10% that is familial. 10 to 20% of that, or if you do the math, 1 to 2% of all ALS is caused by mutations in the gene superoxide dismutase 1, or SOD1. And I'll talk a lot about SOD1 this evening. That overall, approximately 300 to 350 people in the United States with SOD1 related ALS. So I mentioned already, SOD1 ALS is superoxide dismutase 1. It causes 1 to 2% of all ALS. It's a genetic change, so one letter typically that's changed. It's not associated with dementia. This distinguishes it from some other genetic forms of ALS. And there's a toxicity to the neurons, the motor neurons, those cells that connect from the spinal cord or from the brain out to the muscles. That's that toxicity from an abnormal protein that's produced. And tofersen, which I'm going to talk about today, is designed to lower SOD1 protein and is designed to treat ALS for those with the SOD1 mutations. So I understood that this was a relatively lay audience, sophisticated lay audience, but I want to go through DNA, RNA, protein to really help you understand as best as possible the therapy that we're developing. And for the scientists on this webinar, I apologize that this may be somewhat basic for you. So if you think about DNA, I think of this in terms of building a house. You can think about the DNA as a set of instructions. And so this set of instructions is actually kind of hard to use, kind of hard to read. So it needs to be rewritten into RNA, a similar set of instructions, but more like a pamphlet for how to build or maintain, for example, a living room. And you can think about the proteins would be then the stuff in that living room, a chair, or for example, a rug, or perhaps a brick. And then if you think about abnormal genes, there's a change in the genetic code that leads to a change in the RNA, and that leads to the buildup of toxic protein. And so, again, you can think about this DNA as a book or a plan rewritten into RNA, which is a pamphlet for how to develop a space, and then maybe building a brick. But unfortunately, in the setting of the mutation, this brick is abnormal. You don't make the normal brick, you make something that looks misshapen, and then you put that misshapen brick into the house. And at first, things are okay. This is true with ALS. People are fine for 50 years, for example, with some of these genetic mutations. And then things start to go awry, and the nervous system begins to fall apart, or the house begins to fall apart. So that's the way I think about DNA, RNA, protein. And again, we get this change in the genetic code. We have abnormal RNA and a toxic protein. And antisense drugs work at the level of the RNA, and that's what we'll be talking about. The RNA is targeted, and the antisense drugs are a bit like an eraser. They delete, erase, lower the RNA. And by lowering the RNA that's producing this toxic protein, you then eliminate the toxic protein. So I'm going to talk about the phase 3 SOD1 study. This is the study for SOD1 ALS. In that study, SOD1 was lowered, neurofilament was reduced. The primary endpoint was not met, but at the longer term data, and I hope that I'll be able to convince that of you tonight, there's some stabilization and maybe even some improvement, which is unusual for ALS. Here's a bit more on the study design: 72 people on drug, 36 on placebo. And this is showing you the loading dose period, 3 doses in the first month, and then a dose about once a month after that. Tofersen, like other antisense oligonucleotide gene-targeted therapies, don't cross the blood-brain barrier. So tofersen doesn't cross. It needs to be intrathecally delivered, and what's shown here is a spinal column and a needle that can go through the separations in the bones in the back into this space here, if you can see my cursor, which is the intrathecal space, and then the drugs distribute throughout the brain and the spinal cord. It's important to mention the open label extension. So 88% of people chose to participate in the open label extension. That's the period of time after the 28 weeks where everyone goes on drug. And I'll show you that line in some of these studies, open labels where everyone's going on drug. So this is the first piece of data to show you. And I want to orient you to this and several other slides where blue is those that got placebo at the beginning, Red is those who got the drug from the beginning. And then this dashed line down the middle at 28 weeks is when everyone went on drug. And the first thing to note is that the total SOD1 concentration in the cerebrospinal fluid, that's the fluid that bathes the brain spinal cord, was lowered. That's good news. That's exactly what this drug is supposed to do, block the level of the RNA and then block the production of the protein. And you can see that it's lowered. Okay, so how are we going to read out whether this drug is having an effect? One way to look at that is neurofilaments. These are structural proteins. They're biomarkers of nerve injury, and they come in a couple of different flavors, heavy, medium, and light, which is related to just that, heavy, medium, and light in terms of the size of the protein. And they're in this space, the axon. So this is the nerve body, if you can see my pointer. And then this is the axon. That's the connection that goes from the spinal cord or the brain out to the muscles. So that's a connecting piece. And the neurofilaments leak out of this connecting piece. They're structural parts. They leak out when the cell is damaged, and they show up. And they show up in the cerebrospinal fluid, the fluid that bathes the brain and spinal cord, and also in the blood. So we can read that out as a marker of nerve injury. And the neurofilament levels are particularly high in ALS. As you can imagine, they're high in other diseases, for example, Alzheimer's disease and Parkinson's disease and several others here. But they're 2 to 3 times higher in ALS than, for example, in Alzheimer's disease. So particularly high. So why am I focused on neurofilaments? One is that they matter. This is one of literally dozens of figures that show the relationship between the neurofilament level and survival. And I'll walk you through this. So the beginning, there's 100% of people surviving. And then here's the time since that time. And you can look at, for example, out at 4 years. For those with high levels of neurofilament, shown in red, less than 25% of people surviving at 4 years, which of course is not good. Those with a low level of neurofilament, shown in green, more than about 60% of people surviving at 4 years. So high levels or low levels of neurofilament are an indicator of how rapid the disease progression will be. Okay. So the next point to make is that tofersen lowers the levels of neurofilament. And you can see again in blue are those that started out on placebo. Their neurofilament doesn't change until they get out here at 28 weeks, and then their neurofilament goes down. And the group that was on the tofersen or the SOD1 antisense oligonucleotide initially, their neurofilament is lowered. My interpretation of these data, especially in the setting of knowing that neurofilament is so closely tied to progression rates, my interpretation of these data is a substantial slowing of the neurodegenerative disease process. So, does this have an effect on the disease? And to do this, we're looking at the ALS functional rating scale. There are 4 what we call subdomains, so 4 parts focused on movements of the face and the throat, for example, breathing, gross motor or walking, and then fine motor, in particular, using the hands. And at 28 weeks, which was our readout for the primary endpoint, there wasn't much of a difference. There is a difference, but it's not large. This was a bit of an oh no point for us in the setting of the study. We read this out in October 2021. These are all the data we had. We were confused. Neurofilm was lowered, substantial slowing of the neurodegenerative disease process, but yet at 6 months, 28 weeks, not much of a change. But when you begin to look out here at a year and longer now that we've had the opportunity to do, when you look out at a year, you see this group initially on drug doing better than this group initially on placebo, though everybody got drug here, and you see a much bigger difference. These are my favorite data from this published study. I should have mentioned that this was published in September of 2022. And here we're looking at handheld dynamometry. So handheld dynamometry is just a fancy word for muscle strength. We're combining a number of different muscles and comparing it to where was somebody at the beginning of the study, and then how do they progress over time. So it's a measure of strength. And if you look at the group on the drug initially in red, and then look out at 52 weeks, they're clearly doing much better than the group that was on the placebo initially. and they seem to have stabilized. Their strength is no longer going down. And as I look at these data, admittedly, there's some variability here and relatively small numbers. I think that their strength is increasing. And so if you then look at just from the beginning to the end of the study, 52 weeks, who had a strength increasing? Look at the group in red, they got the early start, and about 27% of people had their strength increase over the course of this study. And as I had been presenting these data and talking to local groups and talking to people around the nation who take care of ALS, I asked our neuromuscular group, we're a large group at Washington University, about 178 years of experience taking care of ALS patients. And I asked, so who's seen somebody with ALS get better? And our answer, at least at our institution, is 0. It's not that it never happens across the nation, but it's incredibly rare. And here in this study, 27% of people getting better. I want to share with you some of the comments from the FDA Advisory Committee. The FDA got together in March of 2023 to listen to the public and their opinion about how things were going with this drug tofersen. And this is a 38-year-old woman with SOD1 ALS who says, since that first dose, I'm doing things that I could not do and had accepted that I would never do again. My limp has gone from an obvious disability to barely perceptible. I can go into my office again because I'm able to get up and down the subway stairs. I can shower without my shower chair. Another quote from this FDA Advisory Committee, 43-year-old gentleman, said, I feel like my ALS has slowed down due to taking tofersen. I've noticed my voice has gotten stronger, and having been in a wheelchair for months, I can now walk short distances that I haven't done since before tofersen. This is a gentleman with A5V ALS, a particularly aggressive form of this disease. Most people die in a year or a little over a year. And that's what he's reflecting on here. He says, on the 1-year anniversary of my diagnosis, when I should have died, and that's what his doctors might have told him, I instead kicked a football 47 yards. On the 2nd-year anniversary, I drove a golf ball 275 yards. On the 3-year anniversary, I hit a baseball pitched by my son off the center field fence, then hoisted in the air. That's joy made possible by science. And there are reports from globally from colleagues also sharing their stories about these data. From one of my colleagues at Massachusetts General Hospital, Merit Sokovich. In my 30 years as an ALS physician, this is the first study where I've personally seen people stop progressing and some of them recover function. And then from Pam Shaw, from the participant, I can walk without my poles. I can climb my garden steps, which I haven't been able to do for 2 years. I can write my Christmas cards this year, which I couldn't do last year. There are side effects, and I feel like it is important to mention that they're rare, but there are side effects: myelitis, radiculitis, papilledema, aseptic meningitis, and other adverse reactions. So I do want to call those out. And of course, for anybody with SOD1 ALS listening to this presentation, you would of course want to discuss the risks and benefits with your physician. So some conclusions: lowering of SOD1 lowers neurofilament, and my interpretation is a substantial slowing of the neurodegenerative disease process. At the later time points, there's stabilization of function, breathing, strength. I did show you all those data, but there's also some evidence of improvement, and there are remarkable individual stories. I showed you just a few this evening. There are many more, and many from our own institution. Overall, it's well tolerated, but there are some noteworthy side effects, and I just so this drug was FDA approved in April of 2023, and I'll mention that the drug was then renamed Qalsody. Is the same thing as tofersen. I often refer to it as tofersen because that's a drug I've been studying in clinical trials for the past number of years. Some of the milestones here. This is the first genetic therapy for ALS. The first accelerated approval for ALS. It was the neurofilament was the basis of the approval. It's the first drug to show substantial slowing or improvement in ALS. Other drugs have shown some slowing, but this is showing really substantial slowing and also improvement. It's the first drug to show evidence of clinical improvement in adult onset neurodegenerative disease. Really an impressive study from that point of view. And then some lessons learned about all ALS, and maybe broaden this to lessons learned about neurodegenerative diseases. I think the most profound lesson for me is that ALS is a treatable disorder. I'm not sure that we knew that for sure before this. You'd say, well, you spent 20-something years studying ALS. Of course, you think it's a treatable disorder. Yes, I do. I have always thought that it was a treatable disorder, but this is the study that shows me that it is. Of course, we need the right drug. Tofersen or Qalsody is the right drug for SOD1 ALS. And I think the challenge to researchers like me and around the globe is now to find the right drug for the rest of ALS. This is 1 to 2% of all ALS. And then I have in here ASO delivery to the CSF can be effective. And that's an important statement for all other genetic forms of ALS and for other ALS. Again, that using the same blueprint that we've outlined here can be used for a number of other genetic disorders. And data that I didn't bring in, I kept this talk focused on ALS, but we have really wowing data for Alzheimer's disease showing that an antisense oligonucleotide approach can lower the levels of one of the proteins associated with Alzheimer's disease and have really impressive effects of clearing out aggregated, misfolded, unwanted protein in the brains of people with Alzheimer's disease. So again, this is the first step. And as we think about, again, for this group, that there may be many ways we can use this kind of technology to target genes in the brain and spinal cord to treat neurologic disease. Some of the other points, it may take time to heal. This is something that we are discussing a lot these days in the setting of ALS. Do we need to run a longer trial? How can we see the signals early? How come it took that longer? I'll just mention that neurofilament, I think this is a big deal, and that movement in neurofilament is going to be predictive of success or failure. Though I should be quick to point out that an effective drug may not move neurofilament, and I'm happy to talk about reasons why. And then we need to know more about this drug, know more about the protein neurofilament and what it can be teaching us in the setting of neurodegenerative diseases. There are many, many organizations and many scientists and many people who have been involved in these studies, which started approximately 20 years ago in terms of the first targeting of a gene in the brain or spinal cord using an antisense oligonucleotide technology. I want to in particular call out the study participants, the brave volunteers who make a study like this go, and their caregivers and their families. Without them, of course, we couldn't make these kinds of advances. Also want to recognize the investigators and their hardworking site staff, and the steering committee, in particular, my colleague Merit Sokovich, and also recognize my colleagues at Biogen, Steph Fredette, and Toby Ferguson. And I look forward to taking some questions. And I will stop sharing at this point.
Dr. Miller, that was a spectacular presentation with remarkable data. Thank you. We'll open it up for questions. So Ginger Smith has her hand up. Ginger, please.
Hi. I'm wondering if that can be applied at all to neurodegenerative disease such as Lewy body dementia that I had once, but actually recovered from, rarely, when they removed all my medications when I was in hospice.
Right. So I do think that this is a technique that's broadly applicable, right? I should be careful with the lowering of SOD1 applies for SOD1 ALS, and so you can choose any gene though. You know, for could be alpha-synuclein or something else for Parkinson's or Lewy body or some other gene. And I mentioned already the targeting of protein tau is the name of the protein for Alzheimer's disease. So I think that this can be done for many different neurodegenerative diseases.
Thank you.
There's another question here, Dr. Miller. can you explain the measurement of neurofilament clinically? How's that done?
So neurofilament is a protein that can be measured clinically by a number of different labs throughout the country. So the way that it's done is a blood test. So blood is drawn, that blood is shipped off to a clinical lab, who then measure it by a variety of techniques, typically using an antibody that another protein that binds specifically to neurofilament, recognizes it, and then that gets read out by a number of different ways, number of different machines. I could talk about the different technologies, but I assumed that this was a more generic question about how to use these kinds of measurements.
Thank you. So question here, I have the genetic C9orf72 mutation. Anything to target this mutation?
Yeah. So there was an antisense oligonucleotide trial. In fact, two of them for C9orf72. One was run by Biogen and Ionis Pharmaceuticals. The other was run by Wave Pharmaceuticals. Unfortunately, in that study, neurofilament increased. So the antisense oligonucleotide lowered the levels of the C9orf72 gene product, the C9orf72 RNA. The levels of neurofilament went up, and people got a little bit worse. It wasn't a lot worse, but they got a little bit worse in that study. So both of those groups, and I don't know the exact status of Wave, but I do know from Biogen it was very clear, both of those groups shut down their development of those particular antisense oligonucleotides. And there's a lot of questions to address for that, and I won't be able to go into all of them, but it could be that we targeted the wrong thing. We thought we knew the biology, but maybe we targeted the wrong place, and it's a complicated biology for C9orf72, and so people are kind of back to the drawing board to try to understand how to target C9orf72. And then there's a number of other things coming through for ALS using a similar strategy, but hopefully to treat C9orf72 as well. So I would say stay tuned.
Thank you. There's a question here. Since this is a single-letter DNA coding error, could CRISPR gene editing be used if the anomaly was recognized in utero or at an early age?
Yeah. Just make sure everybody knows what we're talking about. CRISPR is a technology that can change DNA. It's actually really crazy to think about applying this. It's really crazy to think about applying it to cells when it was first done, and then now it's being applied to some of the animal models, literally changing the DNA. CRISPR has an incredible promise as a tool where we may be able to go in and change the genetic problem and correct it. That would be it. That would be the ultimate one and done. We go in and change that gene and fix it. It literally is that kind of technology, CRISPR. So yes, CRISPR could be used to fix SOD1 mutations, most of them which are a single base pair or single change in the genetic code. There's lots and lots of practical challenges with working out how to use CRISPR technology in humans, not the least of which is making sure you don't change a bunch of other DNA. Oh no, it's got to be super specific. If you're going to go in and change your genetic code, you better get that right. And then delivering it to the right spot is also going to be a challenge. And there are groups working on CRISPR technology for C9orf72 in the ALS space and a whole bunch of other genetic disorders. It may be applied first to something in the periphery, taking out blood cells, changing those, putting those back in. Those are the kinds of things I anticipate we'll see first. But I think we're all watching very carefully what is going on with the CRISPR technology and anticipate that it's just a matter of time. It will be in humans, but it's going to be complicated. I'll leave it there.
Yeah. What's the cost of this drug therapy, Dr. Miller?
Yeah. Let's see. So it's $14,000 per dose. The dose is given once a month. So, but I showed you there's a loading dose, And so you get 3 doses in the first month. It roughly works out to a bit more than 200,000 in the first year and a bit less than 200,000 in the subsequent years. There are some different pricing. I'm giving you the approximate from what I understand.
And is this covered by many insurances?
This is brand new.
Yeah.
It just got approved in April. I would
Yeah.
say within the last couple of weeks, got approved to be on formulary at Washington University, right? So just, the wheels turn. And we have the first person at our institution who should be getting, we've been covered, people have been covered with an expanded access program in between the time that the trial ended and now. So people who wanted to be on drug could be on drug, and that's happened. So we didn't stop treating anyone. We were just treating them through this expanded access program. But I think we have the first person that we have lined up to get clinical drug. So the insurance approvals are moving. It should be covered by insurance. I don't anticipate that there'll be major problems. There's a small group of patients with a genetic form of the disease with a therapy that's FDA approved and works. But having said that, that's been true for other disorders and other times. So I think we'll have to see how this goes. So it's going to be, I think at first it's going to be complicated. And for those with SOD1 mutations who are interested in starting this therapy, it may be helpful to be at a large center to start. There are many centers around the United States and Europe who have a lot of experience with intrathecal delivery, and in particular a lot of experience with this drug.
Question, I guess, related to that is, what is the average cost of care for ALS? So while the drug is expensive, what would be the cost shift, especially considered unemployment, less care, et cetera?
I don't have that number for you, unfortunately. I mean, hundreds of thousands would be my guess, but I don't have that sort of cost analysis, and probably should to have this kinds of discussion with people. So thank you for that reminder. Someone's asked me that before, and I haven't dug back in. It's not my type of research, but those numbers are known. I can figure them out.
Question here is, what about the P300 acute quantitative EEG as a marker of neurodegenerative disease?
Yeah, so P300 is one of the evoked potentials, as I understand it. It's been a long time since I thought about P300. And so it's basically stimulation in the periphery with reading out over the brain, that stimulus, which is telling us how well functioning the brain is. It's been used or thought about in the setting of a couple of different neurodegenerative diseases, and I don't remember the details. Dr. Dodick may be able to inform more on that. It's not been used in the setting of ALS, though electrophysiology has been used in the setting of ALS, and one of those is to look at magnetic stimulation of the motor cortex and to read out in the periphery kind of how quickly signals get from the brain to the periphery. And there does seem to be a clear change, I would say, in the electrical properties of the connection between the brain and the peripheral nerves and the muscles. But it's a bit complicated. It's not as simple as a breakdown, but it does indicate that there's probably a change in the electrical properties of the brain and spinal cord that may be very important in ALS. There are some studies looking at this. One therapy that began to explore that. So I think I'd say look for more in that space when those are evoked, other kinds of evoked potential with transcranial magnetic stimulation.
Yeah, I'm aware that there's been, with event-related potentials like N100, P300, N400, work being done in Alzheimer's disease and cognitive impairment. And I myself have been involved in some research looking at event-related potentials in concussion or mild traumatic brain injury. So it appears to be promising there. So as you said earlier, watch for this space. Could be a sensitive biomarker, electrophysiological biomarker for certain diseases. There's a question here about the use of ASOs in spinal muscular atrophy, and are there studies planned for antisense oligonucleotides in other neurodegenerative diseases? Or an asymptomatic ALS with a hereditary, let's say, SOD1?
So there were maybe three questions there.
Yeah.
One is antisense oligos in the setting of spinal muscular atrophy. I mean, the antisense oligos in the setting of spinal muscular atrophy work fantastically well. The name of the drug is nusinersen. It's a splicing change, so you don't want to lower so spinal muscular atrophy is caused by the loss of the SMN1 gene, survival motor neuron gene, SMN1. And if somebody's has lost both of those copies, they typically end up with spinal muscular atrophy, so they've lost it all. Humans, unlike other mammals, by the way, have a backup copy of a gene called SMN2. It looks just like SMN1, and makes the same exact protein, but it's got a flaw in it such that it doesn't construct the pieces of the RNA that make the protein. It doesn't construct it right, splicing change. And so if you use this drug nusinersen, which doesn't lower the levels, that actually just repairs that splicing defect, people make then full length of the protein, make normal amount of protein. And if you get into the kids early enough, it has a dramatic effect. Like it's, for those who take care of spinal muscular atrophy and know what some of those kids should have looked like, it is incredible effect. So yes, antisense oligonucleotides for spinal muscular atrophy. I should be quick to point out that there are two other drugs. One, an oral splicing drug for spinal muscular atrophy, does the same thing as nusinersen, but you take it by mouth. There's obviously some nuances that we could discuss. And then a gene therapy that uses a virus to deliver that gene that's missing. That's called Zolgensma. And that also works very well. So spinal muscular atrophy has three gene targeted therapies, all of which work well. So that was the first question. The other question was about using ASOs broadly for neurodegenerative diseases, like where else are they being used? And the answer is for many, many neurodegenerative diseases. Like this, SOD1 ASO was the first. We did the first in man clinical study, the first ever to put in this kind of drug into the cerebrospinal fluid, and we tiptoed into it. We put in just a little bit. We then redeveloped that ASO, came back, and are now working with the one that we think is quite successful. But other forms of ALS, sporadic ALS, being treated with antisense oligonucleotides as well. For ataxin-2 is one of the genes. Stathmin-2 is another gene that's coming through. I already mentioned tau in Alzheimer's disease, which, again, I'm biased because we developed that drug, but it looks fantastic. Clears out tau in the brains. And, so impressive there. Being considered for Parkinson's disease, alpha-synuclein, a whole host of rare genetic disorders, spinal cord injury. This approach in neurologic disease is in fact enormous at this point. And there was a third question in there that I may have missed.
Are studies being planned for asymptomatic ALS with hereditary?
Oh, yeah.
Yeah.
I probably have some slides on asymptomatic ALS. Do you want me to share screen again? Asymptomatic ALS?
Absolutely.
I'm going to jump in and just share my screen. So one of the things I often mention is about genetic testing, and I just want people to know that there are 2 free genetic testing panels available. Any clinician can order these. I'll also mention that genetic testing should involve genetic counseling. It could be that it's done by the neurologist or physician that's testing you, but you should get genetic counseling, in particular if asymptomatic. So asymptomatic genetic counseling has unique considerations. So really important to get that done with a genetics counselor, for example. Again, it can be done by a clinician, another type of clinician who has a lot of experience. And then let's see. So in terms of following people who are asymptomatic SOD1 gene carriers, for those that qualify, I'd recommend that they are part of this ATLAS study. And Atlas is studying those with SOD1 mutations and following their neurofilament levels. So here's the states. You can probably guess where these places are for the different states, but this is from clinicaltrials.gov. If you're not aware of that as a resource, it's a fantastic resource to find clinical trials. And this is showing you the states, or you can then drill down on the institutions that have this study. Again, people with asymptomatic, follow the neurofilament levels, and if neurofilament goes up, then you either get on drug or placebo. If you become symptomatic, so you develop ALS, you get right on the drug. So this would be the best bet, though not all mutations qualify. And so I mentioned here, you could, if you're in a family, get tested and follow with an asymptomatic gene carrier study. For example, there's one at WashU, at MGH and Columbia. Columbia is called the Families Project. WashU and MGH dials, but really we've combined together, become Prevent ALS. And then University of Miami, Michael Benatar's study as well. And so really following neurofilament, those asymptomatic gene carriers, is what many people do. The details of how this is going to be followed are still not clear, and what the next steps would be, and how quickly would you act on that. Those are all things that are being worked out right now. If you become symptomatic, everyone's going to agree that it's important that you be rapidly evaluated and considered for therapy. And our data that I showed you, I focused on the wowing effects of some people getting better, but if you look at the people who got drugged 6 months later, they're not doing as well. So 6 months made a big difference. And I'll also just note, for neurologists, even those that are quite experienced in the setting of ALS, evaluating somebody for early ALS, a conversion from symptomatic, so asymptomatic to symptomatic, it's challenging. And I'll say it's challenging both for the clinician and for the participant, really hard. And I have a number of people who think they're weak, but we're trying to work it out. It can be quite challenging. Okay, that was my.
Are there any technologies that can detect clinical progression in the absence of overt symptoms that is recognizable by the patient or family? And I'll bring up one which prompted the reason for my question, and that is speech and language. There are platforms out there that can detect subtle changes in speech and language that may be otherwise imperceptible to a patient, family, or even clinician.
Yeah.
So are there objective markers of progression that
yeah. Well, so I'm going to take that in two pieces. I'm going to come back to neurofilament, which I just showed you, which rises 6 months before somebody becomes symptomatic. So if you follow asymptomatic gene carriers and measure their neurofilament with frequency, it does go up before somebody is aware. So that technology, blood biomarkers, is a classic sort of one. I think probably works. It doesn't work perfectly, of course, and we don't have enough numbers to really tell, but I don't think it works quite as well for like C9 or 72 ALS and some other forms of ALS. So it may be tricky to use it across the board. And then your other question is, are there technologies? And we love this idea. So I already mentioned asymptomatic gene carrier studies, Prevent ALS, MGH, WashU, Columbia, and then University of Miami, and for doing their own asymptomatic gene study. But with Prevent ALS, we've begun to explore speech and language. And the speech and language changes in the setting of ALS can be picked up by fancy computer algorithms. And often the analysis software can pick, as you just mentioned, can pick something up before the patient, and I think before the clinicians as well. And so, yes, we're very enthusiastic about speech, and I would say that some of those speech changes and the way they progress in the setting of ALS have now been mapped out by different groups. So I think that's coming through, and I like that. The other things we're considering are just movements, and so simple movements. Sleep is another one that people are using, and I should mention that across a number of different neurodegenerative diseases. Sleep is disturbed, and maybe you could see an early sleep disturbance in someone who's converting to symptomatic with the disease, or even as a way to monitor the disease. So there are lots of these emerging technologies, and again, I particularly, so we're testing all of them because that's what we should do as researchers, but I like speech. Yeah. I think that's going to be super powerful. And I'm on the neurology consult service now, running around the hospital, talking to people who need a neurology consult. And one of the things I tell the residents that I work with is that speech is super important.
Yeah.
They need to get used to analyzing speech and listening for different speech, which in ALS, it sounds different than other disorders. Anyway, I won't go on my whole speech thing, but I think it's a great idea.
Yeah. Thank you. There's a question here about basically epidemiology. You know, how many in the U.S. are affected by ALS in general? And, you know, what triggers or provokes ALS to begin? Like, it's a broad question, but maybe you could tackle that.
Yeah, so the epidemiology, about 15 to 30,000 cases in that range in the United States. 300, 350 people with SOD1 ALS. So, this is SOD1 ALS is a rare form of ALS. When you move to some of the other genetic forms, for example, C9orf72, there are thousands of people with C9orf72 because it's just much bigger population. And then what triggers it? What causes it? I wish we knew. I would say that there's some whispers, from the epidemiology. One of the challenges with epidemiology is that it's hard, that it's difficult to understand the contributions of environment over somebody's 40-year lifetime. For example, how would we know what it was that person was exposed to? We're beginning to look at what broadly considered the expososome, and what are some of the things that might be related to triggering to ALS, or there are some professions that are more related. There are some risk factors. For example, smoking is a twofold increase risk. Of course, if you do the math there, the vast majority of smokers never develop ALS. So it's not a one-to-one. It's just an increased risk. There's probably a genetic part of it. There's what I consider broadly metabolism related. Again, these are older studies, but people who played varsity athletics, slightly increased risk. Lean body habitus, slightly increased risk. People with diabetes tend not to get ALS. People with ALS tend not to get diabetes. So it's not 100% true. These are like broad numbers. Epidemiology, remember, is looking at the kind of broad strokes. People with diabetes do get ALS. There's not any nevers here. So those are some of the epidemiology, some of the triggers. One way that we're looking at triggers is by these asymptomatic gene carrier studies.
Yeah.
I follow a woman who has SOD1 ALS, has SOD1 gene carrier, and her son had ALS and died of ALS. She's like in her 70s. She has an aggressive SOD1 mutation. We double checked. She definitely has a mutation. She's like, well, what should I do? I said, well, whatever you're doing, do the same thing, because you have beat the odds. Like, whatever you eat for breakfast, eat that for breakfast. Whatever you're doing, keep doing it. Because here's someone who could be teaching us what the triggers aren't in a way. And so we're trying to learn from the people who have escaped disease, but who carry one of these really terrible genes. And problem is that there's not enough of them, but we kind of go in with hypotheses and we're trying to figure it out. The other thing I'll say to this and other audiences, if you think let me know. I listen to all the stories. I listen to all the stories from the patients that I see. I'm convinced that some of that will eventually synthesize into something that I can understand about potential causes.
Yeah. There's a question here about whether you see high neurofilament in non-neurodegenerative diseases that have a motor expression like cerebral palsy.
Let's see. So neurofilament is broadly increased in any disorder that leads to damage of axons, any at all. Some other examples include, I mentioned Alzheimer's disease, Parkinson's disease, traumatic brain injury, neurofilament goes through the roof. Spinal cord injury, neurofilament goes through the roof. Some peripheral neuropathies, in particular those that are caused by your body attacking itself, autoimmune neuropathies, neurofilament is high. And so we see a whole bunch of categories of things where neurofilament is increased. Having said that, when I think of cerebral palsy, I think broadly as well, broadly as a birth injury, broadly as something that happened early on in life and is now stable. And so from that point of view, I would expect that neurofilament would not be particularly elevated in that disorder once something has happened. So for example, in the setting of stroke, you might get neurofilament up, but then after a while, once things have stabilized, there's no more neurons being lost, then I think neurofilament would be back down. So it correlates with the with neurons being lost at the time, probably not with the fact that they were lost or damaged in the past.
Yeah. I have a question about, I think we all understand the value of placebo-controlled trials, and in this particular trial that you outlined, it was basically a 7-month double-blind placebo-controlled
Yes.
randomization and then an open-label extension. But the patients, given the prognosis of ALS, 7 months is a long time, and those patients in the open-label extension never caught up. So the question is, couldn't one use, given how tight the correlation is between, elevations and drops in neurofilament light, couldn't one use a biomarker like that to follow patients for 3 months, let's say, and then have an open label extension?
Yeah. So lots of debates about how to run studies, and you've really hit the nail on the head with some of this. I guess in our particular study, the criticism was not that we did it too long on placebo control, was that the criticism really is that we didn't do it long enough. They really should have seen to see the signal, we would have needed to continue the placebo longer, because we didn't see it at 6 months. And we saw some really striking benefits at a year, but the FDA approved our drug on accelerated approval on the neurofilament biomarker. We didn't hit the primary, and so as you know, primaries and those count. And so a lot of discussion, Europeans having, one opinion, and, United States still worth wrestling with it. Most of the trials have been 6 months, and we worry that we've missed a signal. But you mentioned now that we've established that neurofilament can be lowered and that neurofilament lowering is probably important. You could consider running shorter trials. You could consider this, right? You would have and I like the idea, by the way, but this is a Tim Miller opinion, not necessarily an opinion from the broadly from the ALS community, right? Which is, I think we should be running a bunch of drugs through and doing short trials, and then looking for the neurofilament benefit. The downside is that you're going to toss or not promote some drugs that would have had a change or could have had a clinical benefit that you didn't see. So you'll have I guess that would be a false negatives. And I guess you could have some false positive, a drug that affects neurofilament but is really no good for ALS. But I think the likelihood of that is much less. And so then you would screen a bunch of things quickly, and then you say, well, it did lower neurofilament, then do a longer trial, right? And then you have to wrestle with, do you do placebo, and for how long do you do placebo? Do we do the kind of go big or go home trial, which would be, we're going to say like improvement. We're going to key our trial for improvement. Well, that's quite different. If the drug shows improvement, then yeah, okay, it's working. Because in general, we don't get any improvement in the setting of ALS. But even that is harder to define than you'd think, and there is some noise. So anyway, it's a tough topic. I'm absolutely thrilled that we have the opportunity to have the discussion, because this is really one of the first studies to show dramatic lowering of neurofilament. We didn't know that would work.
There's a question here about, was quality of life considered in the study? Were there instruments used? Not surprising, given the sort of anecdotes that you read from the people whose quality of life was dramatically altered. But was that shown statistically between the two groups?
Yes. So that was, and I didn't bring that data. I don't think I have that in a backup slide to show you tonight. But there was big differences between those on placebo and those on drug in terms of the quality of life scales as well. And there were several different quality of lives, the ALS, quality of life scales. The only one where there wasn't as much of a difference and the data were confusing was on the fatigue severity scale, in part because it's a difficult scale. But I think well, I mean, I guess there are other interpretations of that as well, meaning that maybe that was one piece that wasn't as well treated by this drug, or that the drug induced some fatigue. So I think that would be the one quality of life scale where it wasn't as clear. But for the others, the quality of life matched up as a group. And then the other thing is that, of course, not everybody had a fantastic recovery in the setting of this study.
Yeah.
The other thing to mention is there's a bunch of people that just didn't change. A number of people that treated with the drug over the course of years, whose ALS just hasn't changed at all. We've got a guy in St. Louis who's like, "You sure I still have ALS?" Just because he got a little bit better, and then he's just stayed the same. And in his family, typically people die in 5 to 10 years, so it's a slowly progressive form of the disease. So anyway, yes, quality of life was also changed.
Yeah. The final comment here was from one of our audience members who just says, "Thank you. Your passion is evident." And I would say that that's true. This is really groundbreaking. And thank you for the work that you do and will continue to do on behalf of all of us. It's a devastating disease, and it's just so exciting to see this kind of material progress being made. So it's just the beginning, I'm sure. So thank you so much for everything.
Thank you. And I also, I do hope it's just the beginning, that we can broaden this to other neurodegenerative diseases, and of course in other forms of ALS, which, yes, I'm very enthusiastic about doing. So thanks for all the really good questions tonight. What a great discussion, and thanks to the American Brain Foundation for hosting these and putting these on. And Dr. Dodick, a pleasure to meet you. Hope we overlap again sometime soon.
I hope so, Dr. Miller. And thanks everyone for joining us tonight.
Okay, thank you.
Have a great night, everybody.
Thanks. Bye-bye.
Bye-bye.